The Coagulase test is commonly used by microbiologists to assist in the rapid identification of Staphylococcus aureus. Coagulase is a bacterial enzyme that converts fibrinogen to fibrin, resulting in blood clotting. Because bacteria such as Staphylococcus aureus have coagulase bound to their surface, they can distinguish themselves as a blood clot, thus preventing phagocytosis by white blood cells. S. aureus produces two types of coagulase: bound coagulase, also known as clumping factor, for which the slide test is used, and free coagulase, for which the tube test is used. The bound coagulase causes the bacteria to form clumps. The free coagulase causes the soluble fibrinogen in the rabbit plasma to form an insoluble fibrin clot. Hardy Diagnostics product CoaguStaph contains lyophilized rabbit plasma with EDTA, which tests for the production of coagulase. Coagulase is a heat-stable enzyme mainly found in Staphylococcus aureus, and is used to differentiate Staphylococcus aureus from other commonly isolated staphylococci that will be coagulase negative. The Staphylococcus species that are capable of producing coagulase include S. aureus, which is potentially pathogenic in humans, and the animal isolates S. intermedius and S. hyacus, which are generally not found in humans. Rarely, the slide test will produce false negative results with Staph aureus. For this reason, all negative slide tests are recommended to be followed up with a coagulase tube test for confirmation. It is also recommended that the more specific tube test be performed when the specimen is from a sterile site, such as blood or joint fluid, since S. lugdunensis and S. schleiferi can produce false positives on the slide test. First, we will demonstrate how to perform the coagulase slide test. You will need clean glass slides, saline or sterile distilled water, test organism, CoaguStaph, Coagulase Plasma with EDTA, sterile pipettes, inoculating loops, permanent marker. To start the coagulase slide test, have your materials prepared on your disinfected surface. Label your slide with two sections, test and negative control. Reconstitute each vial of CoaguStaph plasma using sterile deionized water. Place a drop of coagulase plasma on your clean, dry glass slide on the side labeled Patient Test. Next, place a drop of distilled water or saline on your slide on the side labeled Negative Control. With a sterile loop or wooden stick, emulsify an amount of the isolated colony being tested into each drop, inoculating the water or saline first. Try to create a smooth suspension. Do not take colonies from a mannitol salt agar plate. Observe for clumping in the coagulase plasma and a homogeneous suspension in the control. Clumps that will not mix uniformly into coagulase plasma represent a positive slide coagulase test and are indicative of S. aureus. A negative reaction is recorded when colonies mix smoothly into solution. Clumping in both the coagulase and negative control indicates that the organism autoagglutinates and is unsuitable for the slide coagulase test. When autoagglutination is observed, the tube coagulase test should be employed as an alternative to the slide agglutination test. Now we will demonstrate how to perform the tube coagulase test. You will need 3 small sterile test tubes labeled Test, Negative Control, and Positive Control, CoaguStaph Coagulase Plasma with EDTA, test organism, sterile pipette, sterile deionized water. Reconstitute each vial of CoaguStaph using sterile deionized water. Aliquot 0.5 ml of reconstituted CoaguStaph into sterile test tubes using a sterile transfer pipette. Store CoaguStaph that will not be used immediately at a temperature 2 to 8 degrees centigrade for up to 30 days. Using a culture that is less than 24 hours old, inoculate the CoaguStaph by emulsifying 1 loopful of bacteria from a non-inhibitory agar plate into the tube of plasma. Incubate the inoculated tubes at 35 to 37 degrees Celsius for 1 to 4 hours. Negative tests at 4 hours should be held at room temperature for a total of 24 hours before reporting results. Read by gently tilting the tube while observing the clotting of plasma. Results should be read at 4 hours. A positive test for coagulase production results in the clotting of the rabbit plasma. Any degree of clotting is a positive test. All negative results after 4 hours should be held at room temperature for a total of 24 hours incubation. Please refer to the instructions for use for the Coagulase Plasma for more complete information. Thank you for watching this Coagulase test demonstration. Hardy Diagnostics is your complete microbiology supplier. Check out our clinical catalogs in the links below. Subscribe to our YouTube channel for more tutorials and product videos. Remember to turn on the notification bell to be reminded of new videos as they are released. Hardy Diagnostics is 100% employee-owned and has been serving microbiologists for over 40 years. To learn more about Hardy Diagnostics and the products we offer, visit us at hardydiagnostics.com.